Dear students, good morning to one and all. Today we are going to learn about mutation. So, mutation means is nothing but is a sudden change of a gene or chromosome from one form to another. So, the term mutant was first introduced by the De Veres. The term mutant referred to the persons who exhibit these inheritable alternations. So, mutation was first discovered by Wright in the year 1791 in male lamp, which had short legs. Laterally, the mutations was reported by Hugo Davis in the year 1900. The Morgan in Drosophila and several others of various organisms. The mutation was coined by the De Veres. Stefanowski stated that mutation is a mistake or misprint in cell divisions. So next we are going to learn mutations and their types. So mutation is a process that produces a gene or chromosomes that differentiate from the wild type. The mutations may result due to change either one stage of chromosomes or genes. Uh, the chromosomes or genes, uh, the changes in the structure and number of chromosomes is called genome mutations. It's otherwise called a chromosomal aberrations. So first we are going to learn chromosomal mutations. The chromosomes contains many genes. The changes in the structure of the chromosomes being about the changes in the number of and arrangement of the genes. These are four types, namely deletions, duplications, inversions, and translocations. So first we are going to learn deletion. Deletion means is a chromosomal aberrations where a segment of the chromosome is lost. Here some genes are lost. So deletion is of two types, namely terminal deletions and intergallery deletions. In terminal deletions, a terminal segment is lost. In intergallery deletions, an intermediate segment of the chromosome is lost. When deletion occurs in one member of homologous chromosomes, a deletions will be produced in the normal homologous chromosomes. In human babies, deletions of segment of chromosomes number 5. A cause of disease is called a chat syndrome. The baby cries like a cat. Is a mentally retreat with a small head. So next we are going to learn duplications. So duplication is a type of the mutation in which a portion of genetic material or chromosomes is duplicate or replicate, resulting in multiple copies of that regions. So duplications produce positions effect. A phenotypic effect are produced by changes in positions of a gene or group genes is called a positions effect. So next we are going to learn inversion mutations. So inversion means the segment is broken in two. A inversion is a chromosome aberrations where a segment of chromosomes breaks and reunites in reverse order. In reverse there is no loss or gain of genes, but the genes are rearranged in reserve orders. Inversions of two types, namely pericentric inversions and paracentric inversions. So pericentric inversion means the centromere is included in the inversed segments. In paracentric inversion means the centromere is not included in the inverted segment. So next we are going to learn translocation mutations. So translocation mutations take place when a portion of chromosomes is relocated. A translocation is a chromosome aberrations where non-homologous chromosomes exchange the segments. Translocations produce a cross-shaped structure during the pairing. Translocation cause positions effect. Translocation alter the linkage groups. So next we are going to learn change the number of chromosomes. Change in the number of chromosomes is called the polyploidy. Ploidy may be due to the loss or gain of chromosomes. A set of gene in the number of chromosome sets. Based on these types, they are divided in two different types. One is aneuploidy, another one is a euploidy. So aneuploidy means a chromosome aberrations where there is a gain or loss of one or more chromosome in a set. In aneuploidy is caused non-disjunctions of chromosomes. It's divided in a three type. One is in monosomy, nullisomy, and trisomy. So monosomy means it's a chromosomal aberrations where one chromosome lost from one pair. For examples, a monosomic Drosophila has 8 minus 1 is equal to 7 chromosomes. It is an aneuploidy. It is produced a woman when egg without an X chromosomes are fused with a sperms containing an X chromosomes. It causes a syndrome is called Turner syndromes. So next we are going to learn nullisomy. So nullisomy means it's a chromosomes aberrations where both chromosomes of pair are lost. It represented by 2n minus n. It is an aneuploidy. A nullisomy is produced by the fusions of gametes having one chromosome's loss. So next we are going to learn trisomy. Tri means 3. Extra on chromosomes added to a pair. It is represented by 2n minus 1. A trisomic individual has an additions chromosomes from the normal number. For example, Drosophila has 8 chromosomes plus 1 chromosomes added is equal to 9 chromosomes. A trisomy man has 46 plus 1 is equal to 47 chromosomes. So trisomy is caused by non-disjunctions. So trisomy 21 man causes a syndrome is called Down syndromes. A trisomic man has 47 chromosomes instead of 46. They are mentally retarded. They have broad face and flat stuffy noses. So next we are going to learn euploidy. So euploidy is a chromosomal aberrations involving a changing in number of chromosome set. It is in two types. One is an haploid, another one is a polyploid. So haploid means the basic set of the chromosomes of any other species. Each chromosome is represented slightly. This is n number. The gametes carry haploid numbers of chromosomes. During the fertilizations, parental chromosomes unite together and fusions the gametes forming diploid number of chromosomes. The next one is a polyploidy. Poly means a lot. It's a conditions which an organism contains more than usual two set of the chromosomes. Such as animals are said to be a polyploid. So polyploid organisms have a 3, a 4 or more set of chromosomes and they are called triploid, tetraploid, pentaploid, hexaploid, heptaploid and so on. So next we are going to learn gene mutation. So gene mutation means it's nothing but the changes in the base sequences of gene is called gene mutations. So mutations produce an alternate gene. The organisms carrying the alternate gene is called the mutant. The processing of producing mutations is called mutagenesis. Mutagenesis classified into the two different types. One is a spontaneous mutations, another one is an induced mutations. So spontaneous mutations means the mutations are naturally called spontaneous mutations. It's due to normal cellular operations are due to the random interactions with the environment. So next we are going to learn point mutations. So point mutations means when a single base pair is altered, the mutation is called point mutations. So point mutations are classified in four types. One is a base of substitutions, insertions, deletion and inversions. So next we are going to learn silent mutations. Silent mutations of three bases in the DNA may result in the substitutions of a new amino acids in the polypeptide chains. When the substituted amino acids is closely related to the original amino acids, mutations has no detectable effect on the phenotypic of the cells. This mutation is called silent mutations. So next we are going to learn missense mutations. So missense mutation means sometimes in a polypeptide chains and amino acids is substituted by another amino acids. This is due to the substitutions of three bases codon in the DNA. This mutation is called missense mutations. So next we are going to learn frame shift mutations. So a mutation that insert or delete a single base will changes the reading frame from the entire sequences. A changes of reading frame is called frame shift mutations. A frame shift mutation shifting the grouping of this bases and changes of code in the amino acids. So next we are going to learn nonsense mutations. Nonsense mutation means sometimes mutations produced a basis sequences that does not code for any amino acids. In such a cases, terminations of synthesis of proteins occurs at the point. This type of mutations is called nonsense mutation or chain terminal mutations. These are three kinds of nonsense mutations on ampher, Orchi and Ophel. Thank you, Ananda.